I'm not convinced that this is the right way to think about neuroscience. So I'm going to start with one of the grandfathers of the philosophy of science, and that is Thomas Kuhn. So in the mid-1960s, Thomas Kuhn wrote a book called The Structure of Scientific Revolutions. Now, he wasn't a philosopher, and in fact, it's a history book, but he wasn't a historian either. He's a physicist. But he became interested in how conceptual change occurred in physics. So he wrote this book. And it immediately, I think, became extremely well regarded. Everybody who was anybody read it. And in fact, I was assigned it as one of my first readings when I attended college way back in Europe. And I remember being horrified when I read it, because one of the things that he was concluding was that there really is no such thing as scientific progress. We change our minds a lot and we have different theoretical frameworks, but the idea that science is somehow marching toward the truth with a capital T, he didn't think was happening. And I was just stunned. I mean, whoa, it changed my world. And I remember going home over winter break and talking to my father about it. My father was a physicist. And I said, Dad, can you believe what I am reading? And even worse, it was taught by a sociologist, whom at the time I thought was like barely a scientist, if that at all. And my dad said to me, why would you think we're going after the truth? What a weird thing. That blew my mind again, because I always thought, you know, we were kind of on the same page where science was concerned. Later, you know, as I learned more, it made sense. He did quantum mechanics, which is kind of unreal anyway. But nonetheless, Thomas Kuhn had a huge influence on me and the trajectory of my career, as well as the way people thought about science and the progress of science ever since. And I think most philosophers of science in some period of their career either embrace Thomas Kuhn or spend their life explaining why Thomas Kuhn is wrong. So here's basically the way Kuhn thinks science goes. So you start with some pre-science phase. It's not really well articulated, but you're kind of diddling around, trying to get things figured out. And from pre-science, you enter into a period of normal science. And when you're in normal science, everyone pretty much agrees what the interesting questions are. They pretty much agree on the methods that you use to answer those questions. And they're kind of little busy bees churning out scientific experiments and publications, all agreeing on the questions and the methods. But what happens as you engage more and more in a particular scientific project and you're trying to develop, let's say, a model of some corner of the universe, that the model starts to change and drift over time as you realize your first impressions were not entirely correct. And as it drifts further and further away from the original conception, he thinks sometimes you end up with this crisis, that the model really doesn't fit the phenomena you're trying to study. And you just can't tweak the model anymore. When that happens, he thinks, we get a model revolution. So someone dreams up a different way of accounting for all the data. But what's important to Kuhn is that these models are incompatible. So you're not adjusting the first model, you're completely changing your mind about whatever it is you're interested in. And then if the new model is any good, you get what's known as a paradigm change. And people talk about paradigm changes all the time now, sort of every aspect of life. And once you've gotten to a new paradigm, then you enter into a new period of normal science where everybody agrees on the new questions you're supposed to be answering, the new methodologies, and we return to being little busy bees, doing our work, getting published, until things continue to accumulate. So if we look at, in more detail, what's going on, what happens when you move from your period of normal science into a period of crisis, you're accumulating anomalies. So in your studies, you're discovering things that just don't fit in the original model. And he argues that there are a lot of sort of non-rational strategies that people use to manage these anomalies, like they ignore them, or they pretend they're not there, or or they blame faulty instrumentation. Sometimes they try to alter the paradigm to fit the new data. But a lot of times, he thinks, we're just so caught up doing our normal science, we're in our groove, that it's really hard to switch, to think about how to accommodate these new data. And then what happens is we get young whippersnappers, young people, who are not so in their cognitive ruts that they can't think about the world in a fresh way, and they realize that all these anomalies that have been accumulated over a period of time add up to really showing that the original model is lousy, and we need to change it into something else. And when we do that, it causes a shift in worldview. Just like I had when I was 18, and I learned that maybe science really isn't pursuing truth with a capital T, but it has other goals and other things that it's trying to do. And when you have this big shift, you really see the world in a fundamentally different way, or at least a corner of the world in a fundamentally different way. And once you do that, there's kind of no going back. You can't see the world through the old paradigm eyes anymore because you realize it's false. Okay, and then, as I said, you adopt this new paradigm, science settles down again, and it becomes routine. So, Kuhn, as I mentioned, was a physicist. The history he was interested in was the history of physics. In the example he discusses quite extensively, and everyone else afterwards, in talking about how to understand this notion of Kuhnian revolution, is the Copernican Revolution. And so you had this notion originally that the Earth was at the center of the universe and the heavenly bodies orbited around the Earth. And then we started accumulating anomalies as our instrumentation got better, and we realized that no, maybe everything wasn't, it just wasn't fitting together. And so we end up with these weird epicycles until we got epicycles upon epicycles, and eventually that whole paradigm kind of collapsed under its own weight. And that's when somebody came up with the idea, maybe we aren't the center of the universe. Maybe we're one of those things that's orbiting around something else. And so then we switched to this idea that the sun was in fact, it wasn't in the middle of the universe, but it was what we orbited around. And so you can see that these two perspectives are really incommensurate with one another. You can't believe the Earth is at the center and the sun is at the center at the same time. And once you switch to the new perspective, then you can start doing more studies on how things orbit around the sun, and that's kind of where we are today. So if we're going to look at neuroscience, I think perhaps one example of how to think about this sort of paradigmatic shift in neuroscience perspective was when we switched to the Neuron Doctrine. And so we had this conflict between Golgi and Cahill over how the brain was structured. And before that, there was an additional conflict where the perspective was the brain is just kind of this undifferentiated mass. Other people thought, no, it's got bits that we can identify. And then Golgi developed a stain that allowed him to see the structure more clearly in the brain, but he clung to the old view that really it's this undifferentiated mass. So he had the reticular theory that everything was kind of, you could see a structure, but it was all connected. There weren't individual bits there. And then Santiago Ramon Cahill argued that no, those little bits are there if you look really carefully, and they're neurons. And neurons are the fundamental unit of the brain. Now, I think we're now at a point where maybe you can see some epicycles developing there. Obviously, I do believe in neurons, but I'm not sure that they're the currency of the brain. Maybe it's groups of neurons, firing patterns. There are all sorts of things that we can talk about that might be fundamental to understanding brain activity. But this, I think, is the type of example that Kuhn had in mind when thinking about how progress occurs in neuroscience, in science in general, and then how progress occurs in neuroscience. Because we would have a hard time doing our neuroscience today if we still bought into the reticular system. Okay, now obviously in the 50 plus years since Kuhn wrote, there have been a lot of challenges to his views. One of them, of course, is that normal science can incrementally tweak theories over time, such that after a period of time you end up with something different, but there's no big conceptual revolution. It's just kind of this incremental change. Another complaint is that the revolutionary science need not be incommensurate with the revolutionary science. The revolutionary science need not be incommensurate with the normal science. And you can even point to Kuhn's pet example when you're talking about the heliocentric and the geocentric views of the universe. I mean, we still understood what the Earth is. We just disagreed about its location. We still understood what we meant by sun. We just disagreed about its location. And so it's not that when you have this conceptual revolution, like everything changes, there are certain overlapping and common themes, and people argue about how important that is. And then other people argue, look, there's some things that we do believe are revolutionary in science that just don't fit this pattern. And one example that's commonly given is the discovery of the structure of DNA, which completely changed the way we think about replication and duplication of genetic information. But there was no huge conceptual revolution, no paradigm shift, people argue, that happened when they came up with the structure of DNA. Okay, recently, John Biffle, who's one of my colleagues in the United States, has argued that maybe this works for physics. He's not a physicist. He doesn't know, and frankly, I don't think he cares. He cares about neuroscience. And he argues that, look, if you look at the history of neuroscience, really what drives change and advancement in neuroscience is not conceptual revolutions, but it's tool development. As we develop new and better tools, we get these big leaps in terms of conceptual progress. And so one example that he points to is when we developed good microelectrodes, then we can implant them in moving creatures and study the reactions of neurons while the animal is behaving. And from that, from the development of these high impedance microelectrodes, we develop sort of the field property theory of neurons, that neurons respond to particular things in the environment in a particular way, and we could demonstrate it using our new tool. And he would argue, going back to the reticular theory versus the neuron doctrine, and this is just my interpretation of him, I've never actually asked him about this, is that really what drove this change was the advent of the silver staining technique, which allowed both Golgi and Cahill to see the structure of neurons. And once they could do that, they'd argue about what it meant. But then when you got electron microscopy, you could really see what's going on in the brain. Then we could see that, yes, there are individual neurons, and that's the way we need to conceptualize the basic structure of the brain. So he would argue it was really the development of the tools that drove this change. It wasn't anything else. Another example, of course, is optogenetics. And so here you can have rapid and reversible manipulation of neurons. Again, it's the same idea that you can see behavioral changes immediately as you change neural activity, which does allow a much more precise mapping of what's going on inside the brain. And so, for example, we've learned how we can implant false memories of conditioned fear using poor, scared mice and rats through lights in their brain. All right. So here's the question. Is our understanding and reconceptualizing of the microbiota in our gut as influencing our central nervous system a revolution? Now, when you do a quick survey of the literature out there, certainly people engaged in this work believe that they're being revolutionary. You can just look at the title. So the Microbiota Revolution, Diet Hypothesis in Light of the Microbiota Revolution, the Emergent Microbiome, a Revolution, the Microbiome Revolution, and the Medicine Revolution of the 21st century. So there's a lot of hype, certainly in the United States, about how this might be forcing a reconceptualization of brain function. So here are the questions that I want to address. The first is, do advances in our understanding of the microbiome constitute a Kuhnian revolution? And if they don't, is that a problem for the neo-Kuhnians? Since Kuhn's dead, he probably doesn't care anymore, but other people who like Kuhn do. Or do the advances in our understanding of the microbiome constitute a Bifflean scientific revolution? Is this being driven by tool use and tool invention? Is this how we should understand it? And if it is not, is this a problem for John Biffle and his graduate students? And in general, what does our understanding of how our understanding of the microbiome has developed, what does it tell us about the philosophy of neuroscience? And ultimately, do we need to change how we understand theories or progress in neuroscience? And I'm using the microbiome as a case study. Okay, so here, I'm not in the United States. In the United States and in Europe, there's a lot of energy and money being thrown at understanding the microbiome. In the United States, we have the National Institute of Health Human Microbiome Project. We have the European Commission's Metagenomics of the Human Intestinal Tract Project, MetaHIT, it's a great name, and Common Fund's Human Microbiome Project. So for those of you who are not familiar with your guts, there is an estimated 10.4 number of microorganisms that live in our guts, which are more than the number of synapses in human adult brains. So there's a lot. And it's not just in our guts, but there's more in our gut than pretty much anywhere else on our bottom. That's 10 times the number of cells in a human body. There's somewhere between 500 and 1,000. This number keeps changing. Maybe it's more. Different species of microorganisms that we lug around with us every day. And there's an estimated 3.3 million different microbial genes in the human GI tract, which is more than 150 times the number of human genes. They estimate this is between like 1 and 2 kilograms that we lug around with us every day. This has a significant impact on our functioning as a biological system. And I think it's only recently that we've really, I think, started to understand the full import of what it means to carry around so many microorganisms inside of our gut. They function there, and they do things to us and with us. Okay, so what we do know is that bacterial colonization of the gut affects the way we mature. It affects our immune system, it affects our endocrine system, it affects our central nervous system. And we know that human infants are almost germ-free when they're in utero, not entirely. And meconium seems to have some bacteria in it. I'm not sure anyone has a good story about what's going on there, but we'll just say they're relatively sterile when they're born. But having, in particular, a vaginal birth distributes microorganisms all over the baby, and they're up and launched. And having a cesarean section distributes fewer and a different distribution of microorganisms. But again, it gets the baby launched. And when you breastfeed a baby, that gives more microorganisms to the infant. And what we know is about age 3, their gut bacterial profile is roughly what you see in our population. I mean, there are variations across individuals, but by about age 3, no matter how you get into the earth, no matter what your mom or your dad feeds you, you end up kind of with the same sort of gut. What we now know is that the bacteria and the distribution of bacteria in our gut affects CNS programming and signaling. In particular, it looks like it can impact our mood, it can impact cognition, it impacts how much we weigh, and it impacts how we experience pain. And this raises the intriguing popular idea that if we modulate our gut microflora, then we might be able to treat central nervous system disorders, right? Just have some yogurt and you'll get over your depression, is the idea. Okay, one of the first things we realized, and by we, I mean other people, not me. They just tell me about this and then I put it on a slide. It's a very, very complicated system, not surprisingly. And there are lots of communication routes between what's going on in our gut and what's going on in our head. So there is a direct neural pathway through our vagus nerve, through our enteric nervous system up to the CNS. And in fact, the place where you have most neurons concentrated outside of the brain is in your gut. There are humoral pathways, so things leak into our juices, and these can sometimes cross the blood-brain barrier, and then that can influence what's going on in the brain. Our immune system is another way that what happens in our gut and our GI tract can influence what's going on in our brain. And then there's a variety of metabolic pathways that are still being explored. So the idea that some people are floating is in the sense that the gut monitors what's going on in the brain, and the brain is monitoring what's going on in the gut. So we're like watching each other, and we do this by synthesizing and recognizing, among other things, the same neurotransmitters. So for example, we have serotonin in our gut. In fact, we've got about 90% of our serotonin lives in our GI tract. The remainder lives in our brain. We obviously know, because we take happy pills, that serotonin is very important for how we experience life. It's also very important for GI motility. It's produced in the gut. They think maybe precursors, tryptophan, can cross the blood-brain barrier and influence what goes on in the brain with respect to serotonin reuptake. This is all theoretical at this point. As far as I know, it hasn't been clearly demonstrated in live creatures yet. Okay, so people have started talking about the microbiota brain-gut axis. This has many different names, but this is just what I'm going to talk about, that this is a new thing, that we need to understand this as one system. The microbiota, the brain, and the gut all work together to achieve common ends. And the idea is when we have homeostasis, when we have a healthy CNS function, we also have healthy gut functioning, and then we have normal behavior, normal cognition, emotion, nociception, pain perception. We're not too inflamed, we're not underinflamed, and our gut microbiota are in balance. But if we have stress or certain types of disease, we see alterations in all of these. We have alterations in behavior, cognition, emotion, pain perception. We have strange levels of inflammation that often don't correlate with the disease state that we're in. And then a lot of times, you know, we get dysbiosis in our intestines. So we get an upset stomach when we're stressed. So the argument is, like, all this we should think of as one system that we need to understand kind of separately from understanding different things in our body. Okay, so here's one example that I will try to walk you through, assuming I can remember all these initials. This is what jet lag does. So the idea is that what is going on in our gut impacts and regulates sickness behavior. And so we all know, I presume, what sickness behavior is. It's what you get when you have the flu. You feel really tired, you feel gross, you don't want to talk to anybody, you don't want anybody to talk to you. I really don't want anyone to touch me. I'm like so antisocial. I'm not hungry, and I'm vaguely depressed. So all this compilation of behaviors and reactions is known as sickness behavior. So one way we think that this happens is our gut gets out of whack for whatever reason. Maybe you have an inflammatory response to some invaders, and as a result your immune system kicks into action, and then we have the microbiotic activated molecular patterns that then release into our immune system. This is from our gut into our immune system, which then releases cytokines, which among other things are precursors for neurotransmitters. They can cross the blood-brain barrier. Of course, they're also out there fighting the battle about whatever infection you have. They impact the microglia and the neurons, particularly in the synaptic areas. It seems to increase the number of serotonin receptors in the neurons, decreases the number of serotonin receptors, increases the amount of serotonin in the synaptic cleft, and as a result, people want to say that you get all of these sort of depressed responses. So here's one proposal, I don't want to say it's been verified in any interesting or significant way, in which we think there might be a direct connection between sickness behavior and what's going on in your gut. A second example, and it's becoming quite well studied now, is using germ-free mice as a model system for understanding, in particular, the stress response. So one thing that's very interesting about germ-free mice, and these are mice who are not only raised in a sterile environment, but they are birthed using cesarean section in a sterile environment. They have no microbiota. They're just clean as a whistle. Do you need to leave? Right here. That's fine. So now all the good stuff will happen. And so what we find with germ-free mice is they don't exhibit the same stress responses as germ-laden mice. Even when you have germ-laden mice that have no illnesses or parasites or anything with them. So they're really clean, but they have microbiota. And the two big responses that you see is the germ-free mice exhibit really intense exploratory behavior. So they'll go out in ledges, they'll go out in the light, whereas most mice prefer to be safe and securely tucked away in the dark. So they don't seem to have as much anxiety relative to germ-laden mice. But I have to say, now that I've spent about a month reading all of these studies, the data actually seem to be a little bit inconsistent, and there's some germ-free mice under some examples where they seem to exhibit greater stress, not less stress. And so, for example, if you restrain a mouse, that can be very stressful, apparently, to the mouse, which I understand. I don't think I'd want to be restrained myself. But the germ-free mice have an overreaction relative to normal mice. So it's not, you don't get consistent responses with the light box where they seem to prefer the light. There's some cases where it seems like they actually prefer the dark more. But nonetheless, it does seem to be the case that if mice lack microbiota for their whole lives, they respond differently to the world. Now, if you take these germ-free mice and you give them mice microbiota, particularly if you do it young enough in their development, they develop normal mouse stress behavior. So this was a big aha moment when people realized that there is, in fact, this connection between what's going on in the GI tract and what's going on in the brain. And we can manipulate it by manipulating the microbiota within our model system. And of course, now the big assumption is that what is true in mice is true in humans. A second area that's very popular in the United States, makes me a little bit nervous, is the alleged connection between autism spectrum disorder and the microbiome. Now, it is true that a lot of patients diagnosed with autism or autism spectrum disorder do have GI tract malfunctions. But I don't think it's actually clear why they have them, because a lot of people on the spectrum also have very unusual eating habits. Quite often, they don't like fruits, they don't like vegetables, they don't like carbs, they don't like meat. Actually, they don't really like much of anything, and they pick up 2 or 3 things that they're willing to eat, and that's it. And so it's not surprising in some sense that they would have trouble in their stomach or a different profile of microbiota. Now, it is the case, and it's also the case that the more unusual their microflora in their gut, the more severe the symptoms, and vice versa. What's not clear, I think, is the causal influences, right? Do they have an unusual gut because they have autism spectrum disorder, and that influences how they consume food, or do they have autism because they have an unusual gut, and then that also influences the food they pick, so it becomes this cycle. Now, in the United States, they've come up with these studies that show that if you are breastfed, then you're much less likely to end up being diagnosed with autism spectrum disorder. Therefore, everybody should breastfeed their babies. And the implication, of course, if you don't breastfeed your baby, you're a bad mom, and you're the one who caused your kid to have autism. I myself am not really persuaded by this argument, in particular because we're so US-centric in the United States. We forget the rest of the world, in particular places like South America, where virtually everyone is now bottle fed. There's very little breastfeeding going on. They're trying to reverse that trend, but the diagnoses of autism spectrum disorder are much, much less in South America than you find in the United States. So I'm doubtful how much you breastfeed your baby is ultimately going to make much of a difference. But there's some good questions that could be raised about the brain microbiome link to explain maybe some cases of autism spectrum disorder, or maybe the severity of symptoms, or maybe some things can be done to relieve some of the symptomatology of autism spectrum disorder. And there's also a discussion about where the relevant microbes that babies need. Is it the vagina? Is it from the gut? Is it from the mouth? The teeth? I mean, who knows? But it does seem to be the case, at least with mouse models of autism, if you change the distribution of the microflora in the mice, then you get a reduction of the symptomatology of autism spectrum disorder. So at least for mice, there does seem to be a connection. There is a burgeoning cottage industry in the United States of giving your kid some sort of prebiotic or probiotic drink, and maybe that will help relieve some of the symptoms associated with autism spectrum disorder.